cut it, cut it, cut it, cut it, cut it, cut it.
We're going to be discussing a very important tool called Type 2S restriction enzymes. You're probably already familiar with the functions of a restriction enzyme, which cleave DNA at specific points within a DNA sequence. This is a very useful tool to us as researchers because they let us manipulate and edit DNA for purposes such as DNA fragmentation and gene cloning. You may be wondering, where do restriction enzymes come from anyway? How are they made? Restriction enzymes are typically harvested from bacteria or archaea, and they are named after the microorganisms that they come from, such as EcoR1, which comes from E. coli. There are very few different types of restriction enzymes that differ in a way that they recognize and cleave different sites.
Type 2P restriction enzymes are the most common. The P stands for palindromic. In type 2P restriction enzymes, the part of the enzyme that recognizes the cutting site and the part of the enzyme that cleaves the DNA are part of the same single domain. Type 2C restriction enzymes contain 3 domains, one for recognition, one for cleavage, and one for methylation. The C stands for combined because the enzyme combines cleavage and modification into one enzyme.
Hi. So lastly, there is type 2S, which is what we will be focusing on today. The S stands for shifted cleavage. Type 2S enzymes have 2 different domains, one that recognizes the sequence and one that cuts. The unique thing about type 2S is that it cuts at a different location from the recognition sequence. For example, this is the recognition sequence in the type 2S restriction enzyme BAY1. You may be wondering what those numbers mean. The 1015 means that for the sense strand, there must be 10 N bases, and for the bottom strand, 15 N bases. The enzyme will recognize the base sequence shown here. The Y means it will recognize any pyrimidine. The 12/7 means that the enzyme will cut the top strand 12 bases after the recognition sequence, and the bottom strand 7 bases after the recognition sequence. This leaves sticky ends with a 3' overhang that is 5 bases long. As you can see, the cleavage is shifted from the recognition sequence. The unique benefit of this is that it allows researchers to create custom overhangs that are specific to the DNA that is cut, unlike other restriction enzymes that create the same overhang every time they cut.
You need to cut it.
In nature, restriction enzymes are typically used as cellular self-defense. Restriction enzymes evolved to target sites in viral DNA to prevent viral replication. Restriction enzymes were first investigated in the 1950s by researchers Salvador Luria and company. Viruses grown in the same host bacteria as they previously grew in were seen to propagate effectively. This was because viruses have managed to methylate all the restriction sites in its genome that the host could target. However, if viruses were transferred to a new host, most grew poorly. When transferred to a new strain, viruses lack site-specific methylation to protect them from the restriction enzymes contained by the new host. The new bacterial restriction enzymes would outcompete the methylation transfer proteins produced by the virus to destroy the viral genome. It wasn't until the late 1960s that restriction enzymes were actually recognized for their potential applications in biotechnology. Werner Arber first quantified restriction enzymes in 1960s by looking at the phenomenon host-controlled restriction of bacteriophages. Hamilton Smith then verified Arber's hypothesis and published 2 independent classical papers. Dan Nathans furthered this work by discussing possible future applications of restriction enzyme modification.
You need to cut it.
One limitation of type 2 restriction enzymes is that the creation of long DNA sequences via forced cloning. Since a given enzyme can have many restriction sites in a long segment of DNA, lining together DNA sequences longer than 1,000 base pairs without digesting it at points in the middle of the sequence becomes problematic. Researchers have begun experimenting with hybridizing type 2 restriction enzymes with homonucleases. These nucleases will hopefully seek out highly specific sequences near the restriction site of the interest, hopefully targeting the restriction enzyme to a specific restriction site on the DNA.
Cut it, 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 cut it. Them bricks is way too high, you need to cut it. Your price is way too high, you need to cut it. Cut it, 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 cut it. Them bricks is way too high, you need to cut it. Your price is way too high, you need to cut it. cut it, 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 cut it. Them bricks is way too high, you need to cut it. Your price is way too high, you need to cut it. Cut it, 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 cut it. Them bricks is way too high, you need to cut it. Your price is way too high, you need to cut it.